Good morning, everybody, on this beautiful, almost spring morning. It is my distinct pleasure to be able to introduce Dr. Roberto Bolli. Dr. Kalra is in an airplane right now, so didn't feel, even though we have telemedicine, didn't feel he could do that on the airplane. So we all know Dr. Bolli, but I'm going to go through some of his past. He was a graduate, and I'll say it wrong, at the University of Perugia, Italy. He, as many people did that have achieved great success in research, first did a research fellowship at National Institute of Health, I believe, and then clinical fellowship at Baylor College of Medicine. He was recruited then to Baylor and stayed there for a little more than a decade, achieving professor and tenure in just 11 years. We recruited him here in 1994 as the chief of the Division of Cardiovascular Medicine, and he served in that capacity for 26 years until March 2020, when he decided to focus the rest of his career on his research. He still serves here as the director of the Institute of Molecular Cardiology. He's a distinguished university scholar and a Jewish Hospital Distinguished Chair. While you probably don't know this, in his entire time being here, he's maintained grant funding to the tune of $120 million, which brings $40 million of indirect assets to the university. He has 469 papers. He's given 347, so I guess today 348 lectures, but he's done so internationally as well as nationally. He's taught and mentored many fellows that have gone on to attain being division chiefs and department chairs, and all have had esteemed careers under his tutelage. Roberto has received numerous prestigious awards and honors, not just in this country, but worldwide. His current research focus, and his research focus really throughout his career, has been on mechanisms for myocardial injury, ischemia, reperfusion, nitric oxide, etc., with the goal to use these understood mechanisms to develop cardioprotective strategies. So today we're going to learn a lot about what he's done in the last decade or so on cell therapy for heart disease. Roberto?
All right, thank you very much, Chris, and good morning to everyone. So today I will discuss my last 20 years of work on cell therapy and most importantly provide a current status on this new revolutionary therapy. I want to say at the outset that we do not have any cell therapy approved yet for the treatment of cardiovascular disease. In other countries, there have been some forms of cell therapy approved, but not yet in this country. But I believe that cell therapy will become part of our therapeutic armamentarium in the next few years in the United States. And the purpose of my talk is to give you an update of where we are in the translation of this very promising therapy from the clinical realm to the clinical arena. So I am probably the only person in this field who does not have a company or a financial interest, and I'm just doing this out of scientific interest. I don't need to remind you, I'm sure you're aware that heart disease is the #1 killer in the United States, getting worse in 2020 versus 2019. And stroke is #5. Kentucky has the dubious honor of being at the very top of the list of states that are plagued by cardiovascular disease, as you can see here. About 29% of deaths in Kentucky are caused by heart disease and 6% by stroke. So 1/3 of patients die in Kentucky from cardiovascular disease. And this is the problem that causes heart failure in many patients. This is the heart of a patient who had a massive anteroseptal infarction reperfused with streptokinase. You can see the dark hemorrhage in the heart. Now, nowadays we can keep some of these patients alive, but what happens, they go on to develop remodeling and ventricular dysfunction and heart failure, and they live for a few months or years a miserable life, and then they die. There are about 6 million Americans with heart failure. The number keeps increasing as the population ages. 600,000 new cases every year in the United States, and the 5-year survival for patients with heart failure is 50%. It's worse than many cancers. So this is really a public health problem of major proportions. We don't have any really good treatment for heart failure. We have a lot of drugs that can make patients feel better, maybe live a few months longer, but really they don't have a material impact on the prognosis, hence the need for new treatments such as cell therapy. So by the way, what I'm going to present today is summarized for those of you who are interested in this review. It's a very detailed review. I spent a lot of time writing it. That was published last year in Cardiovascular Research. Okay, a couple of minutes to discuss the concept of stem cells. So a stem cell is a cell that has two fundamental properties. Number one, it is able to differentiate into a mature cells, one or more mature cells, I should say. And number two, it is self-renewing. There are two types of stem cells: the pluripotent stem cells, which include the embryonic stem cells obtained from the inner mass of the blastocyst, and the induced pluripotent stem cells, which are obtained from just about any type of adult cell, which can be made regress to an embryonic state and then differentiate again into a different type of cell. The pluripotent stem cells have the ability to generate basically any type of cell in our body. So you can take an embryonic or an induced pluripotent stem cells and produce a brain, heart, lung, kidney, whatever. The adult stem cells, on the other hand, are multipotent, meaning that they can only differentiate into a limited number of adult cells, one or two. For example, the mesenchymal stem cells in the bone marrow can produce cells of mesodermal lineage, meaning adipocytes, bone, cartilage, fibrous tissue. The hematopoietic stem cells can produce blood, but they cannot produce other tissues, and so on. So the adult cells have more limited differentiation ability. The most important thing we have learned in the area of cell therapy in the last 20 years is how these cells work. Initially, it was thought that when you transplant a stem cell into a disease organ, let's say a disease heart, or for that matter, any organ, kidney, brain, or whatever, that cell will differentiate into the cells of that organ. So if you take a mesenchymal stem cells, you put it in the heart, it was thought that that cell becomes a cardiac cell. But then we have learned that this is not the case, and I will show this a little more in a little more detail in just a few minutes. These cells do not differentiate. In fact, they die off very quickly after transplantation. They do not survive. So this differentiation hypothesis is now abandoned, and now we have learned that all cells, including the pluripotent stem cells, not just the adult cells, but even the embryonic or the induced pluripotent stem cells, they work by paracrine effects, meaning that they secrete a panoply of mediators, microRNAs, bioactive lipids, cytokines, growth factors, and others, which then exert favorable influences on the surrounding tissues and produce the therapeutic effect. So this has been the single most important conceptual advance in the field of cell therapy in the last 2 decades. I said the pluripotent stem cells include the embryonic cells and the induced pluripotent stem cells. The embryonic cells were initially I mean like 20 years ago, they thought to be a revolution in medicine that would really provide a cure for many diseases by essentially regenerating any tissue of the body. Well, that is now no longer true, and I will show you why these embryonic cells basically, in my opinion, and I think it's shared now by many other investigators, are not likely to become a clinical therapy. First of all, there are obvious ethical and regulatory issues because these cells require the destruction of a human embryo. Secondly, these cells are known to produce tumors. And in fact, the way you determine whether a cell is an embryonic stem cell is by determining whether they are capable of producing teratomas. Now, the last thing you want to do in a patient with heart disease is to produce a tumor in their heart. They require also prolonged culture to produce billions of cells, and that, of course, is a setup for genetic abnormalities, which then, in turn, can cause tumorigenicity. Because they belong to a different organism, the recipient must be immunosuppressed for life, and that in itself is a disease. But perhaps most importantly is that now we have learned that after you transplant embryonic stem cells into the heart, these cells do not survive as it was originally thought. They do not differentiate into new heart tissue, and after 1 month they're basically gone. That's a paper that was published in Circ Research a few years ago, the largest study ever done of human embryonic stem cells in primates. These were monkeys done in China. And basically you can see that after transplanting these human embryonic stem cell derived myocytes into the heart of these monkeys, after 3 months and a half there were no cells left. They were all gone. Furthermore, in these studies in monkeys, the embryonic cells produce ventricular tachycardia and fibrillation, which obviously is not very desirable. And finally, with the advent of induced pluripotent stem cells, which are essentially the same as embryonic stem cells, it is not clear why one needs to use the embryonic stem cells, which cause, as I said, rejection and require immunosuppression. So for all of these reasons, I believe that the embryonic stem cell based therapies are not going to be used in cardiovascular medicine. On the other hand, the induced pluripotent stem cells may have a clinical application, although at the moment there are no clinical trials using these cells in the United States. There may be in the future. One of the problems with these iPSCs is that they are tumorigenic, just like the embryonic cells. The other problem is they're very expensive to make it. I mean, to produce iPSCs from each patient is financially not a very good model in terms of making this a widespread therapy for widespread use. So I'm going to focus the rest of the talk on the adult stem cells, and just give you a quick update of where we are in this ongoing revolution. A few minutes about the preclinical experience, what we have learned. First of all, it is now unquestionable that many different cell types are beneficial to the injured or failing heart. By beneficial, I mean that they improve left ventricular function. This cannot be questioned. This has been shown in hundreds of papers. This is a picture of 2 rat hearts where we produced an infarction and then we harvested the heart 1 year later. On the left, you see a control heart with a very thin scar, huge aneurysm, you would say. And in the right side, you see a treated heart treated with mesenchymal stromal cells where the scar is much thicker and much smaller. So you can see much less remodeling after. This is 1 year after a single injection of these adult cells. And when we measure ejection fraction, again, let's just focus on the right panel here. 12 months after treatment, the red bars are higher than the yellow, meaning the treated rats are doing better than the controls. This is 1 year after a single injection of these mesenchymal cells. And we have published literally tens of papers in different models, mice and pigs, showing the same thing. This is an echo of a pig where we produce an infarction, and then 3 months later, repeated the echo, you see the heart is dilated, the ejection fraction has dropped from 73 to 50. 1 month after administration of cardiac progenitor cells, the ejection fraction is even worse, it's 44. So I'm sorry, this is a control. So 1 month after the injection of vehicle, the ejection fraction is actually worse, so which is the natural history of ischemic cardiomyopathy. This is a treated pig that received cardiac progenitor cells, in which you see the improvement in ejection fraction from 47 to 54 1 month after treatment. The anterior wall here moves a little better than before the injection of cells. And we are also using MRI to measure ventricular function and volumes, as well as to measure the size of the scar using gadolinium injection and hyperenhancement, as you can see here in the bottom panel. So using MRI, we see the same results as we do using echoes, improvement in ventricular function after administration of mesenchymal stromal cells in these pigs with ischemic cardiomyopathy. So that the cells are beneficial can no longer be questioned. But there are other issues. First of all, the term stem cells is not appropriate in many cases, because the cells we are using are not really stem cells in the sense I defined them earlier. So I don't like to use the term stem. I rather just call them cells, because in many cases we don't know that they're really stem cells. We have also learned that the transplanted cells, regardless of the type, it doesn't matter which type you're using, bone marrow, CD34 positive cells in the blood, cardiac progenitor cells, adipose cells, it doesn't matter. Any cell type does not engraft and does not form new cardiac cells. Therefore, the phrase cardiac regeneration should be avoided. We are not really regenerating the heart, because we are not producing new myocytes. We are repairing the heart, meaning we are improving the function and the structure of the heart, but we are not producing new tissue. This is just a study where we show that after we injected the c-KIT positive cells in mice with an infarction, by 35 days they were all gone. Either whether we injected them intramyocardially with a needle or intracoronarily, either way, after 1 month, there were no cells left around. And that has been shown over and over in many other models. So, if the cells don't survive, how do they work? Well, what we learned is that all transplanted cells, and this includes, by the way, the embryonic stem cells as well, work via paracrine or perhaps even endocrine mechanism, which means releasing non-coding RNAs, proteins such as growth factors and cytokines, lipids, extracellular vesicles, etc. We don't know yet what exactly the mediators are. There are literally hundreds of things produced by these cells. These cells are factories that release really a panoply of mediators in the surrounding tissue, and it's going to be, I think it's going to be really difficult to identify the mediators responsible for their beneficial effects. And the mechanism by which these mediators work is also unknown. The most popular hypothesis is that they inhibit inflammation in the heart, which is a factor in heart failure, but they may also have antifibrotic effect, anti-apoptotic or angiogenic effects. For example, the bone marrow mesenchymal stromal cells or mesenchymal stem cells are known to modulate neutrophils, macrophages, dendritic cells, B and T cells. So basically the entire immune system, and suppress inflammation, which is known to be a major factor in chronic heart failure. By reducing inflammation, it is thought that these cells will improve the course of heart failure. Now let's move on to the clinical realm that is more relevant to this talk. What have we learned in the last 2 decades? A number of cells have been used in clinical trials listed here. The cells listed in blue are those that are still viable today as a therapeutic option. And they include the bone marrow mesenchymal stromal cells, which are probably the first ones that will become a clinical therapy in patients with heart disease, in my opinion. But there are others, the CD34 cells, the c-KIT-positive cardiac cells, neonatal cardiac mesenchymal cells have shown very exciting results in children with single ventricles, adipose-derived mesenchymal cells, and, as I said, the induced pluripotent cells. Now, these are the 2 major types that are closest to become a clinical reality, the bone marrow mesenchymal stromal cells and the adipose-derived mesenchymal stromal cells. They're basically the same cell type. One is harvested from the bone marrow and the other one from the adipose tissue. And I want to go over a few studies that support the utility of these cells. So DREAM-HF is a phase 3 trial just concluded a year ago, presented in abstract form but not yet published. About 600 patients with heart failure were given bone marrow mesenchymal stromal cells, and there was a reduction in major adverse cardiac events, and I'll show that in a slide in a little bit. So this trial is going to be very important in bringing cell therapy close to becoming a reality. The MSCHF trial was done in Denmark. A very exciting study was a phase 2 trial in patients with ischemic heart failure, ischemic cardiomyopathy. These are the results showing a highly significant improvement in ejection fraction at either 6 months or 12 months after treatment in the treated group, and no improvement, actually a deterioration in the placebo-treated group. So this was a very exciting study. CONCERT-HF was published a few months ago, and we were part of this phase 2 trial. And in fact, we were the #1 enrolling center among the various centers involved. It was a phase 2 study of autologous mesenchymal stromal cells and c-KIT-positive cardiac cells, alone or in combination, in patients with ischemic heart failure. This was published in the European Journal of Heart Failure, as I said, a few months ago. So, CONCERT was a phase 2 randomized, double-blind, placebo-controlled, multi-center trial that was sponsored by the National Heart, Lung and Blood Institute Cardiovascular Cell Therapy Research Network, or CCTRN, and the goal was to investigate the feasibility, safety, and efficacy of autologous bone marrow mesenchymal stromal cells and cardiac c-KIT-positive cells in patients with heart failure caused by ischemic cardiomyopathy. This is arguably the most rigorous clinical trial of cell therapy for heart disease ever conducted. It took us 4 to 5 years just to get started. In other words, just to go through all the regulatory approval, the quality assurance, and the clearance with the NHLBI, the IRB, the DSMB, the Food and Drug Administration, and so on. Basically, it was a 10-year effort by 7 centers, 2 cores, a biorepository, a GMP cell manufacturing facility in Miami, a data coordinating center, and of course the National Heart, Lung and Blood Institute. Truly an enormous effort. This is the organization of the trial. You can see we were participating there in conjunction with the Texas Heart Institute, University of Florida, Minneapolis Heart Institute, the University of Miami, Stanford, and Indiana University. We enrolled 125 patients. The ejection fraction was 28.6 at baseline, and the scar size was 19% of the left ventricle. All of these patients obviously had an infarction, and they were all in class 2, mostly in class 2, New York class 2, or some in class 3. They were randomized 1:1 to 1:1 to 4 groups, either the combination of the bone marrow MSCs and the cardiac CPCs, or MSCs alone, CPCs alone, or placebo. These cells were injected transendocardially by putting a catheter in the left ventricular cavity using the NOGA mapping system. This is an example of the NOGA map, which basically tells us which regions of the heart are viable in blue or purple, and which of them are dead in red. The dots show where the injections were made, about 10-15 injections in each patient, and they were done basically in the border zone between the between the scar and the viable tissue. So the results in terms of clinical outcome were very exciting. This slide shows the heart failure major adverse cardiac events, or MACE. The incidence over a follow-up of a year and a half, I would say, average, was about 28% in control patients. These were placebo-treated patients. But in patients receiving the cardiac c-KIT positive cells, only 6%, 2 out of 31, developed MACE. And in patients receiving the combination of the cardiac CPCs and the bone marrow mesenchymal stromal cells in yellow, also only about 8% received developed MACE. So the administration of a single injection of either CPCs alone or CPCs plus MSCs significantly reduced the incidence of MACE over the next 12 to 16 months. When we broke down the 3 components of MACE, death, so what are MACE? MACE are death or from any cause, hospitalization for worsening heart failure, and exacerbation of heart failure that does not require hospitalization. The major driver of this beneficial effect of cell therapy was a reduction in hospitalization for heart failure, which was about 80% reduced in patients treated with CPCs alone or in combination with MSCs. Truly a remarkable effect. So the Kaplan-Meier curve showed that the number of MACE continues to escalate with time for a year and a half in placebo patients, but in patients treated with CPCs, the cardiac c-KIT positive cells in red, or the combination of CPCs and MSC in blue, there is a marked reduction in MACE that is sustained all the way to the end of the study. The hazard ratio was 0.2 for CPC alone and 0.26 for MSCs plus CPCs. So in other words, an 80% reduction in MACE with cell therapy. The quality of life was measured using the Minnesota Living with Heart Failure questionnaire, a lot of variability, but nevertheless there was a reduction at both 6 months and 12 months in the score, which means an improvement in quality of life. The lower the score, the better quality of life, with the administration of the combination of MSCs and CPCs. On the other hand, we did not see any effect of cell therapy on ejection fraction, left ventricular volumes, scar size, maximal oxygen consumption, the 6-minute walking distance, and the plasma levels of the BNP, which raises the intriguing question of the mechanism, and this we'll get back in just a second. But I guess the conclusion of this trial is that in patients with chronic ischemic heart failure, which are already on maximal guideline-driven therapy, I remind you, all of these patients are taking the best possible medical therapy available today for heart failure. So in these patients on maximal guideline-driven therapy, a single administration of autologous CPCs or MSCs has measurable beneficial effects over the ensuing 12 months. Meaning a reduction in hospitalization for heart failure with CPCs and an improvement in quality of life with the MSCs. The best results were obtained by combining the 2 cell types, MSCs and CPCs, as I just showed you. So this may be a way to go in the future. Rather than relying on a single cell type, it may be advantageous to combine them and have a greater effect. And neither CPCs nor MSCs improved left ventricular function or reduced scar size. By the way, this result obtained with concert is not the only trial to produce this effect. In other words, there are 2 other trials. Let me go to DREAM-HF first, which I mentioned earlier. This is not yet published, but the results were presented at the HA meeting last November. In this phase 3 trial, there was a significant 30% reduction in MACE with the administration of bone marrow mesenchymal stromal cells. And another trial published in Lancet a few years ago with also bone marrow MSCs showed a 37 reduction in MACE. So we now have three placebo-controlled randomized double-blind studies showing a significant reduction in clinical events in MACE, major adverse cardiac events, with cell therapy regardless of the change in ventricular function. I think this is very exciting because obviously patients don't care about ejection fraction, right? All they care is whether they can stay home without going to the hospital or they can have a better quality of life. So if we can improve their clinical outcome with cell therapy, I think there would be really a major advance in the treatment of heart failure. Now, what is the mechanism for this? Because it's puzzling. Why are these patients not getting into the hospital and why are they doing better, even though the ventricular function is not improved and the scar size is not reduced? So the short answer is that, of course, we don't know yet. That's going to require a lot of work. But what we have found recently, I'm doing now this study as follow-up to CONCERT, we are measuring the plasma levels of pro and anti-inflammatory cytokines in these patients before and after therapy. And these are some preliminary data I wanted to share with you where we measured a number of cytokines. I'll just show you 4 of them. IL-1 beta, which is the prototype of the pro-inflammatory cytokines. IL-2 is also pro-inflammatory. IL-6 is also pro-inflammatory, and TNF-alpha. So these are major pro-inflammatory cytokines. And in all cases, you can see that at 180 days after treatment, there is a decrease in the plasma levels of these cytokines in patients receiving cell therapy compared to the controls, placebo patients, which are in green. So in other words, this is evidence that cell therapy does what I told you earlier. It reduces the systemic inflammatory state associated with heart failure, and this could conceivably be a major mechanism by which cell therapy improves the course of heart failure by reducing the inflammation that makes the heart failure get worse and worse over time. So we are still working on this data. It's very expensive to measure these cytokines, by the way, and we are trying to complete the analysis, but we don't have all the numbers yet. Another study that we have done here at the University of Louisville is Seneca, which is a study of allogeneic mesenchymal stromal cells in patients with anthracycline-induced cardiomyopathy. This was published in JACC Cardio-Oncology also a few months ago. The first study ever of cell therapy in this population. These are patients who survived cancer, were treated with anthracyclines, and then a few years later developed cardiomyopathy and heart failure. It was a phase 1 study because, as I said, this was the first study ever in this population. So it was not really powered to measure efficacy. It was was designed to assess safety and feasibility. Most patients were females, and most of them had either breast cancer or lymphomas, and then developed the anthracycline-induced cardiomyopathy, which, by the way, has a worse prognosis than other type of heart failure, and there is really no treatment for these patients. Many of them are very young. At 12 months after the injection of the bone marrow allogeneic cells, despite the very small group sizes, you can see we're looking at 12, 14 patients per group, really very small sizes. There was a significant improvement in the 6-minute walking distance and in the quality of life as assessed by the Minnesota score. All the other numbers also were in favor of cell therapy but did not achieve significance. So these data, and by the way, there was no significant adverse effect. So these data are very exciting, and I think they lay the groundwork for a larger phase 2 trial to determine the efficacy of cell therapy in this very unfortunate patient population. Now, perhaps the most important thing I can show to support the utility of cell therapy is the meta-analysis of clinical trials of cell therapy in patients with heart failure, which was published in Circ Research a few years back, 1,500 patients. And to make a long story short, everything looks favorable in terms of showing the efficacy of heart failure. So the ejection fraction improves on average 4.6 units in cell therapy treated patients versus placebos. The rehospitalization is reduced by about 60%. The even mortality, incredibly, even mortality is reduced in this meta-analysis by half. New York Heart Association class is reduced, and the 6-minute walking distance is improved, and the quality of life is also improved. So everything looks, everything points to a beneficial effect of cell therapy in patients with heart failure in this meta-analysis. So what have we learned clinically on the use of cell therapy for heart disease? First and foremost, administration of adult cells is safe. I did not mention any of this earlier, but there has not been a single instance of a patient that had a major adverse event that could be ascribed to the cell type itself. Some patients had complications because of the delivery of cells, like cardiac perforation when the catheter perforated the heart, but no complications that were caused by the cells in themselves. Secondly, I don't have time to go over this, but there have been many trials of cell therapy in patients with STEMI, and basically they do not show a beneficial effect. I believe STEMI is not a proper clinical target for cell therapy. There are very promising data in patients with refractory angina. Again, I don't have time to go over them, but there are several trials showing reduction in angina, improvement in exercise tolerance, and even myocardial perfusion by giving CD34 positive cells. And as I just showed you, very promising data in chronic heart failure. In fact, in heart failure, the randomized double-blind placebo-controlled phase 2 trials I showed you suggest beneficial effect of MSCs as late, and also CPCs, as late as 1 year after treatment. And the thing that's remarkable is that we have a signal of efficacy despite the fact that we still don't know a lot of things about these about how to use these cells. We don't know, first of all, what is the best cell type, the best dose, the best route. Is it intramyocardial, intracoronary, intravenous, and the frequency? Can we repeat cell therapy and get a larger effect? I believe we can. We've actually done a lot of studies in animals, but clinically this remains unclear. So the frequency of administration is another major issue that has not been resolved. So what is needed now is to move the field forward, is to conduct rigorous, well-designed phase 3 trials to conclusively address the efficacy. DREAM-HF so far is the only phase 3 trial that has been conducted, but we need more. And I believe once we have more phase 3 trials, this will become a clinical therapy. It is important to have an open mind, to avoid preconceived notion, and of course let ourselves be guided by the evidence in assessing the efficacy of cell therapy. I want to conclude just a couple of slides to let the audience be aware of some very exciting therapy for heart failure that's not cell therapy. This would be better defined as gene therapy. So this is the use of cell cycle genes in combination, 4 of them in combination, called the 4F or 4 factors, that are delivered to the heart using an adenovirus. This research is spearheaded by Dr. Tamer Mohammed that we were fortunate to recruit here a few years ago, and is doing a fantastic job and very exciting research. He published this paper in Cell in 2018, and it made it to the cover of Cell. You can see the 4 factors, CDK4, CDK1, CCNB, and the CCND, which basically what happens when these genes are transferred to the heart, they make myocytes re-enter the cell cycle and divide. Myocytes are terminally differentiated. They do not divide, which is the reason why after a heart attack we have a scar rather than new tissue. But when we transfect them with these genes, they re-enter the cell cycle and they make new myocytes. So unlike cell therapy, gene therapy with these factors actually regenerates the heart because produces new cardiac myocytes. And that's obviously very exciting. So this is a study that Tamer just published in Circulation a few weeks ago in our pig model, showing that when we injected these 4 factors in the heart of pigs with ischemic cardiomyopathy, and then we measure ejection fraction, we see after a few weeks, I think it was 4 weeks, if I remember, we see an improvement in ejection fraction shown here, whereas in controls there is no improvement, and a reduction in scar size, which implies regeneration of the heart in just a few weeks of treatment with gene therapy. So this is very exciting. We also do MRI in these pigs, and we show the same thing I just illustrated graphically, which is the improvement in cardiac function. On the top, you can see a control pig where the ejection fraction actually dropped from 26 to 20%. And at the bottom, a treated pig where the ejection fraction improved by 10 points, 24 to 33% after the administration of the 4 factors. We are now planning to continue to do longer-term studies in pigs to see if the effect persists beyond 1 month. And if so, we will then plan a phase 1 clinical trial using this in patients with ischemic cardiomyopathy. And as I said, this would be really very, very exciting because no one so far has been able to regenerate cardiac tissue. So I want to acknowledge all the colleagues that have worked very hard to generate the data I showed you and many other data over the last 26 years. And thank you very much for your attention.
Thank you so much, Dr. Bolli, and Dr. Kruger, we'll let you start off.
Well, first, for the young investigators in the audience, you can see that all the effort and time it takes into bringing science to the bedside for the benefit of all of us, and how important it is to have folks invest such time and energy. It takes a lot of effort. It takes a team effort. Takes groups of people working together and collaborating. I had a technical question, Roberto, and the technical question is, since cells traffic to surface receptors, is it really necessary to inject directly into the myocardium near the scar? Would it be, is anyone just injecting them peripherally? And I know this just because I know you can inject cancer cells in a rat's arm and injure the other arm, and the cancer cells will traffic to the injured area and develop, an implantation at that site. So I'm just technically, it might be easier. I'm just asking, do we really have to inject it directly into the myocardium?
So that's a great question, actually. Until recently, it was thought that cells must be delivered to the heart in order to be helpful. In other words, that they will engraft, they will become cardiac myocytes, and obviously you must put them in the heart, because if you inject them intravenously, they will not reach the heart. Well, as I mentioned, we now know that cells do not engraft. Even when you inject them with a needle inside the heart muscle, they die within a few weeks. So we did studies to see what if we inject cells intravenously. If the cells work by paracrine mechanism, by releasing factors yet to be identified, is it possible that they may release these factors even though they're not in the heart? So we inject them in IV, and most of them obviously go to the lung. Some of them go to the spleen or the liver or other organs. Very, very few get to the heart. And even with IV injection, we still see a beneficial effect on cardiac function. And that's one of the grand proposals that I've been submitting to both the NIH and the Department of Defense, was to do a phase 1 trial of intravenous cell therapy rather than intramyocardial, because obviously intravenous, you can repeat it as many times as you like. You can do it anywhere, even as an outpatient. It will be really very easy to do on a massive, widespread scale. So the answer to your question is that yes, we have evidence that intravenous therapy is beneficial. There are many studies in animals showing that, already published. And that would be a game changer in terms of making this therapy widely available to every patient with heart failure. And I think that's one area that needs to be investigated further.
All right, Dr. Bolli, you had a question in the chat area, actually two. I'll ask the first one. Is from Dr. Singh over in the Department of Physiology. He wanted to ask about the future of xenotransplantation.
Yeah, I presume this is driven by that patient that received a porcine heart. By the way, I read that he died yesterday, I think, the patient, after a few weeks. So I'm not an expert on xenotransplantation. It is an immunologic problem. I simply don't know how much the issue of rejection has been addressed by genetically modifying these pigs. So I'm sorry I cannot give you a very informed answer because I'm not working in this area, but I think it's going to be really tough to eliminate rejection, even if you genetically modify the pig, just because it's a different species. Maybe I'm wrong.
All right. Another question. This is from Dr. Robert Emmons from our oncology, hematology, and the bone marrow area section. He wanted to know, can you comment on the FAP CAR T therapy at the University of Pennsylvania compared to your results?
The CAR T, yeah, so the CAR T therapy is obviously very exciting in cancer. That therapy is a form of cell therapy and gene therapy, right? Because I believe, and again, I'm not an expert, but believe what they do, they take T cells and they transfect them with a chimeric antigen using gene therapy, and then they reinject them into the same patient. So that therapy is very, very effective in some tumors. In the heart, that has not been done clinically. It has been done in animals in the sense that many groups, including our own, have taken a cell type, let's say bone marrow mesenchymal stromal cells, and enhanced that cell, made it more powerful by transfecting it with genes such hemoxygenase 1 or superoxide dismutase or other genes that enhance the ability of the cell to survive once it's transplanted. And we have shown, so if we take these super cells, let's call them super cells, that have been genetically modified, they live longer than the natural cells, but eventually they also disappear. So I'm not too excited about doing this in the heart because the advantage is probably incremental. It's possible though that there may be new genetic modifications we may introduce in our cell therapy that would make these cells much more powerful, and certainly that needs to be investigated. But again, it's a different issue than the T cells that are transfected with chimeric antigens.
But Dr. Bolli, I would just mention that the New England Journal article and their subsequent Nature articles have shown effective delivery to sites of injury because of targeting with the CAR T cells of various genetics, including transient mRNA, which have now been proven to decrease scar and increase physiologic function. So they've been very successful in showing targeted genetic modification and regeneration of the heart. So is that compared to your studies?
Yeah, I am skeptical about those results. I know it's one group, I think it's the Dr. Epstein at the University of Pennsylvania. So it's one paper that came out. Let's see if it is reproduced. I don't know. A lot of stuff gets published and then cannot be reproduced. So let's see what happens. So far, there is one report using that approach you mentioned. And let's wait and see. I think it's a little early to pass a judgment yet.
All right, Dr. Bolli, I have a question. One more question from the chat area is from Dr. Philip May, one of our community doctors. He wants to know, do your patients receive any type of special physical exercise?
No. Some of them undergo rehabilitation, cardiac rehab, I believe. But no, we don't. Certainly in our trials, we did not include any such thing. We test their functional capacity with a very simple test. One is the 6-minute walking distance, right? So we let them walk in the corridor for 6 minutes, see how far they can go, and then we measure the maximal oxygen consumption on the treadmill, and that's kind of a standard treadmill test, but we don't have any specialized protocols for rehabilitation.
All right, and that's the last from the chat area. If anyone has any more questions, if you want to post it in the chat area, if you want to unmute and ask, I think Dr. Bolli may have covered all the bases, Dr. Kruger. So
That's again certainly exciting, and it takes a long time to get there. But it sounds like in the last several years there have been some pretty significant cumulative advancements. So we'll be looking forward to seeing Dr. Bolli, what you and Dr. Tamer and others do in this area for the next several years. Probably there will be. It seems to me like it's reaching a pinnacle where you know which kind of cells to use, and they're actually showing cardiac regeneration, and so in some of them, or as you mentioned, cardioprotection. So sounds like it's reaching a nice time. You mentioned that Europe has already approved this as therapy. So
yeah, for example, the CD34 positive cells are used in the Netherlands to treat patients with refractory angina. So these are cells that are angiogenic. They stimulate formation of new blood vessels. And you know, the refractory angina is a big problem. There are, I think, at least 1 million patients in America that have refractory angina, meaning these are patients that have tried everything, right? They've tried bypass surgery, they tried coronary angioplasty, of course, medical therapy, and they still have angina, and they cannot be revascularized either with PCI or with bypass surgery. So in these patients, there are several trials in the US and Europe showing that the injection of CD34 positive cells improves the vascularization of the myocardium, the blood flow, as well as the exercise tolerance of these patients. These cells are very safe. They don't really have any significant adverse effect. In the Netherlands, and I believe even in Korea, these cells are already being approved for the treatment of refractory angina. So hopefully, you know, this will happen in our country as well. But that's an example of where other countries are a little ahead of us in this field.
okay, well, the hour is approached, and Thank you very much, everyone, for your attention. Thank you, Dr. Bolli, for walking us through a very interesting travels through heart cell therapy. I think that was very comprehensive, and I really appreciate it. I learned a lot.
Thank you very much. Thank you, Dr. Kruger.
Thanks, Dr. Bolli, for nice update.
Thank you. Bye-bye.